Yeah. Okay, let's get started. Let me start sharing my screen. Apparently, it's just something I just learned. I have to start the recording before I started sharing the screen. Since after I started sharing the screen, the record button disappeared. Okay, okay, let's get it started. For the third lecture of the ChIP-seq analysis. So first, I would like to ask as a question to the students. Have you guys already started the assignment for the ChIP-seq section? And which question have you finished so far? And do you have any questions about the getting the data and get the data running? Yeah, you can feel free to let me know if you encounter any problems, any issues when getting the, including getting the data and get the software installed, and any question during the, when processing the data. And you can post the questions either on Slack or email me, or post it on the Colab. So yeah, so this is the third lecture of ChIP-seq analysis. And we will focus on the downstream analysis, data integration, and give you some information about online resources for you to get an idea. So a brief overview of what we've already covered for the last two lectures. So we start from the raw sequence reads data that directly come out of the sequencing machine, and the entire computational analysis of ChIP-seq data start from aligning, assessing the sequence reads quality, aligning the sequence reads to the reference genome to get the aligned reads, and all the way including several quality control measurement, and get the informative mapped reads. And we can use MAX2 as a peak calling tool to identify peaks that are the genomic regions with an enrichment of sequence reads. So now we obtained the max identified peak information that telling the genomic regions that have a transcription factor binding or enriched for histone marks. So now we will do some downstream analysis to interpret these peaks we identified. So for downstream analysis, we'll cover these few topics: DNA sequence at the peaks, namely DNA sequence motif discovery. And for annotation of the peaks. So if we know the genomic location of all the peaks, what can we do about it? What other information can we learn from this information of peaks? And also integration with other omics data and for functional analysis. So I will briefly go through several tools or methods that can be used for this type of analysis. And for a practical reason, I probably will not go into the technical details of the underlying methods and models of these tools, but I just want to give an idea of what each tool can do, and what. So for motif discovery, the ultimate goal is to identify the specific sequences that underlying these transcription factor binding sites. These specific DNA sequences usually represents the DNA sequences that the DNA binding domain of the transcription factor can recognize, and is usually specific to different groups or different categories of different families of DNA binding domains that represents different groups of transcription factors. So of course, for a ChIP-seq experiment, we already know what transcription factor we are targeting at. So for most of the transcription factors, we already know the information of their DNA binding domain and their specific DNA sequence motif that this transcription factor is recognizing. But for ChIP-seq, if we have a ChIP-seq data, and especially we identify the peaks, we will still do motif discovery. So there are several reasons for that. So number one is, this is of course another layer of quality control. So if we chip a known transcription factor, if we want to make sure that the peaks identified from the ChIP-seq data are indeed the transcription factor, the binding sites of that transcription factor, then we should assume that the known sequence motif of that transcription factor should be able to be identified from the peak regions. That means that we indeed identified the real binding sites of the region. And the second point is that although we know the sequence binding DNA sequence motifs for most transcription factors in the canonical cases, so there are several cases that a transcription factor might bind to a non-canonical motif or even an unknown DNA sequence motif. So that is something that for ChIP-seq experiment can directly tell us, because we believe that we generally believe that the binding sites or the peaks identified from ChIP-seq experiment are the real DNA protein interaction sites. Even those specific regions do not have the canonical binding motif. And number three is that we can also find the motif for we may also be able to find the DNA motif for other transcription factors in the peak region. So that could indicate that these transcription factors that we chipped have some cofactors. So that will be a discovery method for identifying potential cofactors or other information that associated with these the binding sites with the transcription factor that we chipped for. So we usually use a position weight matrix to represent a DNA sequence motif. So this type of sequence motif logo we can frequently see in the DNA motif analysis or a lot of the DNA sequences. So intuitively the sequence logo of this type of plot is a graphic representation of a position weight matrix. So a position weight matrix is a quantity or a matrix to quantify a sequence motif. So here is an example of how to define, how to calculate a position weight matrix, the PWM. Say, for example, if we do a ChIP-seq experiment and identify a bunch of binding sites, and then if for very simplified information, if we know that our transcription factor can be interacting with these several DNA fragments, the sequences. So the position weight matrix is to represent or to measure the shared or the common information located in these different sequences. So the way to do is that we have all of those sequences, and we just first generated this count matrix, meaning that for all of these sequences in the candidate sequences, starting with each base pair, each position, we count the number of the 4 different nucleotides, A, C, G, and T, and so on and so forth for each and every of these positions. And then we simply convert it into a fraction, into the fraction of the 4 nucleotides. Say, for example, in the first position, the 30% of them are A, 20% of them C, and so on and so forth. As for in this case, for the 4th position, all of them are G. So 100% of them are G. And the next position, 100% of them are all T. So, so this is the position weighted matrix, the PWM matrix. In order to graphically display or to visualize the information from the position weight matrix, we use this sequence logo to plot this position weight matrix. So there are 2 level or 2 layers of information in position and the sequence logo. So one is that in each position, the height of each letters, each of the 4 nucleotide letters, is proportional to the relative percentage of the of the occurrence of each of the nucleotides. So as we can see, in the 7th position, 70%, 70% are A, so A is higher than the other 3 other letters. As for these 2 positions, of course, well, all of them are G or T. And the second layer of information that are represented in the sequence position in the sequence logo is the information content, or the information content contained in this each region, in each position. So the information content is defined as this. So because this is a DNA sequence, we have only 4 nucleotides. So 4 nucleotides contains 2 bits of information, which means that in the you use 2 bits of binary numbers can represent all the information in this one of the 4 possible nucleotides. And then the H information here is the Shannon entropy, defined as the frequency of each basis as the basis at each position, and times the log of this frequency as this definition of Shannon entropy. It's also an a way of calculating the information content. So, which means that if the more likely that one specific nucleotide is present at a specific location, the more information content we have from this position weight matrix. So that's will give a total height of this sequence logo. So that's how we generate this sequence logo. So not every tool, motif discovery tool, is using the same definition of sequence logo that they generated. So many of them that do not include this content information, content, but simply represents everything as the using the relative fraction of the 4 nucleotides and represents at the relative heights. So in that case, the sequence logo will be all have the same height. So several commonly used tools for sequence motif discovery. I'll just briefly go through them. So one of the most used and early developed motif discovery tool is MIM. So currently MIM has already, the team has developed a series of different methods with the focus of sequence discovery, motif discovery. And the key, the core function of MIM is a de novo DNA sequence motif discovery. So which means that they can look for the enriched or represented, highly represented motifs from a series of or bunch of DNA sequences without the prior knowledge of what existing, what known DNA motifs are for known transcription factors. So in this case, the input of MIM for the motif, for de novo motif discovery, is that we do need the raw DNA sequences. Or the MIM can also take in other type of sequence data like RNA sequence or even protein sequences. So they can identify, perform de novo motif discovery, identify the enriched motifs in the motif patterns from these series of sequences. And so that's the MIM ChIP, including MIM ChIP is from a list of regions or sequences from the peak regions we identified from ChIP-seq experiments. And then after we use MIM ChIP to find those de novo motifs, we can compare this identified de novo motif with a motifs from a databases which contains many known motifs. So for known motifs for known transcription factors. And in that case, we can compare the motifs we identified from our data with the known motifs in the database. They will tell you that which of the de novo identified motifs from our data belong to which transcription factor. So then we can find this the results as a aligned motif or a matched motif with the known information. So of course, this is just a typical or one example of many available tools, available functions in the MIM suites. So you're welcome to try and take a look at some other useful tools in the MIM suites. Say, for example, for FIMO is a tool to scan if we have a PWM, if we have a known motif, they can tell you from the bunch of the input sequences which positions or which locations have that motif enriched in your input sequences. So, for example, if we have the ChIP-seq peaks, we want to know in each peak regions where does our interested motif located. So we can use FIMO. And another motif discovery tool is HOMER. HOMER is specifically developed for ChIP and ChIP-seq data analysis. So I won't go to the details of HOMER, but you can browse the website of HOMER. So just to give the credits, I think HOMER is developed in the UCSD, led by the group of Christopher Glass. And nowadays, HOMER also include more tools for not only for motif discovery, but also for integrative analysis of ChIP-seq data with other type of data, including comparing your own ChIP-seq data with other existing known transcription factor binding sites. So these are for motif analysis. So next, for annotation, or for to annotate the peaks identified from the genome, one tool that is very useful is GREAT. So GREAT is developed by Jill, I forgot how to pronounce his name. Yeah. So GREAT. Jill Bangerano. Jill Bangerano group developed GREAT in, I think, published in 2010. It's similar to a gene ontology analysis. Instead of a usual gene ontology analysis in which you input a bunch of genes, a list of genes, and they can compare the genes with known databases with annotation of what functional group of all genes is. So GREAT can take in a set of genomic regions, such as the transcription factor binding sites or a bunch of ChIP-seq peaks, and they can compare your input regions and give you the functional annotation of all the regions that from your input compare with the known information from the databases that they collected, and not only tell you that so the one information that they can give is that how far are the regions, are the peaks in your input, how far are they from a known transcription start site or TSS that can give you an information of what percentage of the peaks in your ChIP-seq data are located close to a transcription start site, which means they're close to a promoter, or they are located in the distal regions. They're far from any transcription start regions. And also they can give you a list of annotations of what possible transcription factors are also bind in the same regions from your ChIP-seq peaks, and also what are likely functions of these regions, especially for regulatory regions, including annotations. Any questions so far? Okay, so so I should mention this. So GREAT, I think GREAT has both a web interface in which you can directly upload the data, the peak data, through this website, and then directly get the analysis. And I think MIM has both a Python package that you can install and also have a online web-based interface that you can upload to the website to do the analysis. In my impression, Homer is primarily a Python package that you need to install and run through command line. So if you are more familiar with R, and there is a useful tool in R bioconductor called ChIP-seq. It's a they integrated several many existing functions to annotate, compare, and visualize ChIP-seq data. So that's an R and bioconductor package that you can install through bioconductor, and you can simply follow the instructions in the ChIP-seq tool to perform all the analysis that it can generate, including telling you where in the genomic regions that your peak enriched, and even generated several plots for visualization. Will give a summarized summary idea of what your ChIP-seq data look like. So this one is R package. So next, I'm gonna introduce several online resources related to ChIP-seq data analysis that you might find useful. So the first one I want to talk about is Galaxy. So Galaxy is a integration or integrative web-based analysis platform for a lot of genomic data, not only for ChIP-seq. So this is a kind of an infrastructure. So they have this, they build up this. Any question? Or someone? No, that's okay. So they build up this web-based platform so that the community can install or can upload not only upload their own data and to perform analysis on this platform, but they can also install their interested bioinformatics tools on their on this platform to perform the analysis. So this includes pretty much all the tools that I covered in the previous lectures, including FASTQ data quality control and the sequence alignment and the peak calling using several different read mapping sequence alignment algorithms or several different peak calling tools. So let's take a look at the interface of Galaxy. Yeah, so I wanted to mention that James Taylor is one of the early developers or one of the co-founders of the Galaxy platform. He tragically passed away last week. And he's currently a professor at Johns Hopkins. And yeah, so we'll recognize his contributions, recognize his founding contributions in the entire Galaxy platform. So, so they have the interface of so all Galaxy platforms are similar. So there are 3 different sections. So one section is the tools menu, this on the left panel. So they have all the different tools and that are currently available. For example, for FASTQ analysis, FASTQ, they have different type of QC measurement, QC measurement for sequence data, and for mapping, for example. They have different type of sequence aligners for sequence mapping, including, for example, BIOC8145 that we mentioned. So instead of using BIOC8145 in the command line base on our server, you can upload the data that we are interested and then perform the analysis in this web interface. So the bottleneck of this is that in case that the data set is very large, it may take long to upload, and the analysis, because all the analysis is done at their web server, it may take a long time, or it may have a relatively long queue that you need to wait for. So in addition to download the raw data and upload to the platform, Galaxy also can accept direct data transfer from a existing database like NCBI. SRA is the short reads archive that have most of the next generation sequencing data sets as a data repository. So they have BIOC8145, as you can see here, ChIP-seq data. They also have ChIP-seq peak callings, including MAX and ChIP-seq for broad peak calling, and also several other tools for MAX. So I think this MAX is version 1, and we are more frequently use MAX 2 to call peaks. So just give a few examples. So once you submit a job, they will put in the queue and run the analysis, and then once you get the results, the output data will be listed, just piled in this right data directory as your own working directory. So Galaxy is a very easy to use, straightforward web-based analysis platform that I encourage you to give it a try. So next, I want to mention Galaxy, another interactive analysis platform that designed specifically for ChIP-seq analysis is called the Systrom. So Systrom analysis pipeline is, well, the infrastructure is based on the Galaxy's Galaxy platform. So you can see the interface looks very similar to Galaxy. Well, it is indeed just the Galaxy platform as the infrastructure. So for Galaxy, they have the similar interface, and they have the tools built in there are specifically focused for ChIP-seq analysis. Like you upload the data, and to call peaks, and to perform their list of, in the integrative analysis section, there are a list of different useful tools for data annotation and generating plots, as well as discovering motifs. Okay, so now I'm gonna briefly go over several online data resources. So first, ENCODE. So ENCODE is a collaborative consortium led by NIH for the past over 10 years. It stands for Encyclopedia of DNA Elements. And at the pilot start stage, in the pilot project, they still use ChIP-seq to generate data sets for DNA transcription factor binding sites or genomide profiling. And later on, most of the data they generated is using ChIP-seq. So all of the data in ENCODE are publicly available. So that's the goal of the consortium, is to provide these data resources to the community so that they can use. So they contain primarily, early stage, they primarily contains cell lines from human and several other model systems. And recently, they also include more primary tissues in their analysis. So for every cell type, either cell line or primary tissue, they generate their different type of multi-omics profiles, including transcription factor ChIP-seq and histone modification ChIP-seq, as well as their transcriptome data like RNA-seq and other chromatin data. So they have tens of thousands of data sets that cover a lot of different transcription factors, histone marks, and in different cell types. But as we know, a collaborative consortium is only one part of the efforts from the community that generates the data. That besides the consortium, every lab around the world who study genomics are generating different type of data, including ChIP-seq data. So the Systrom Data Browser is another databases that not only include the ENCODE data, but they collected all the available published ChIP-seq data. They collected from the GEO, the Gene Expression Omnibus, which is one of, I think, the single largest data repository that contains all the public data, ChIP-seq data that are published in a paper that we are required to submit the raw data to a public data repository so that the data can be accessible to everyone. So Systrom Data Browser is a database developed by Shirley Liu for the past few years. And they includes primarily focus on ChIP-seq data in human and mouse, these two species. And they collected over nearly 1,000 different transcription factors and histone modifications in all the cell type, primary cell and cell lines available in the public domain that we can find. So we can take a look at the Systrom Data Browser. So when when you generated a in your further studies, if you want to generate a ChIP-seq data for some transcription factor or histone modification in the system that you're interested, you might want to first check what publicly available published the data already published in there in the similar system or in the similar transcription factor or for the factor that you are interested. So that's Systrom Data Browser is a good resource to look at. Say, for example, if we are interested in the androgen receptor AR in human, so we can click this for AR. So then they list all the, we can see we have 19 pages of ChIP-seq data sets for AR in all different cell types or biological sources. So if we take it, for example, if we only focus on one cell line, prostate cancer cell line, LINCAP, click AR again. So there are also 8 pages of AR ChIP-seq data in LINCAP cell line that published by different groups and published in different papers, and they may have under different biological condition and several treatments. But each of them represents a AR ChIP-seq data sets in LINCAP cell line. So a more useful function in this Systrom Data Browser is that they not only give you the data sets, they also have the quality control of these data sets, which is a very important component when you access or when you want to reuse the data. So the quality control that includes 6 different quality control measurement. So green light means that they pass the QC, and the red means that they have some issue with each QC measurement. So these quality control, so once we click one data, they will list all the detailed information of these data sets. Say under what treatment is this biological sample is, and what's the original data that they identified. They have a data accession ID, and in human, and published in which paper, including their PubMed ID, and its AR factor, and all the biological conditions of these data sets. And for each data set, you can directly visualize the data, visualize the BW profile of data using 2 different genome browsers. And you can download the already analyzed peaks or BW bigwig file, or the predicted putative targets of the genes. And they, as I mentioned, they give some quality control measurements. So the 6 measurements, I think we've covered most of them. So includes the sequence quality, the raw sequence medium quality score. I think it's directly from FastQC analysis, and the mapping quality, that's the unique macro ratio. So what percentage of the raw sequence reads are uniquely mapped to the reference genome. And they have the PCR bottleneck coefficient, PBC, to measure the level of redundance, and the number of identified peak. So in this case, the number of identified peak is not very good. So they under standard parameters, they can only identify 378 peaks from this data. So that could be an issue. So that's marked as a red here. And also, we can use a higher look at how many peaks are with a high fold enrichment as another QC measurement. So use a fold enrichment as at least a 10, or fold enrichment at least a 20. So we can see there are very few peaks that have a fold enrichment of at least 20. And FRIP score, and we mentioned that last time, a fraction of reads in peaks, so 0.2% is an issue. Usually it should be over 1%. I mean, over 1 or 2% to several percent. And they tell you the percentage of peaks in different regions in the genome, including promoter, exon, intron, and intergenic regions. And the percentage of the top peaks in the union DNase hypersensitive sites are, we mentioned last time too, that how many of these peaks, how many of those peaks are located in the known regulatory regions in the human genome. So usually a working ChIP-seq data for a known transcription factor, most of the peaks should be in the existing known regulatory regions in the whole genome. So in this case, it's good. It's good to have a high percentage, but part of the reason is because they have too few peaks. So which in this case, it may tell that the experiment still works, that they identified are indeed AR binding sites, but for some reason they are just could be because the ChIP experimental efficiency, but they didn't pull down enough binding sites from the data. And the phase count conservation score plot shown here is to look at the sequence conservation of the peak regions. So as I mentioned last time, for a known transcription factor center, their binding sites, the center of the binding sites usually contains the DNA sequence motif and usually should be conserved in terms of the DNA sequence across evolution. So that's give you all the different quality scores that you can browse and analyze. So as we can see here, among many of these published data, they all really have a various quality of the data. So the quality or impact factor of the paper that the data set is published does not usually does not necessarily have a high correlation with the quality of the data. So that's a take-home message from browsing a lot of public data. So public data is useful for your studies as always, and so that's the ChIP-seq Systrom data browser. So using this a lot of publicly available data, one can develop further bioinformatics tools to study the transcription factors or to study gene regulation for the systems that you're interested.
Changzi?
Yes.
Yeah, I just like to make one point that's just worth noting. I think this is fantastic resource, and I love the way you went through all the QC. I just think it's worth noting, and you're well aware of this, you've just got to be mindful with that one thing about the number of peaks. You've got, the thing to be aware of is look at the complexity of the motif. So for example, and I can't off the top of my head, reel off the AR one. I think either Changzi or Mike could probably, but so for example, when a motif is very complex, like P53. In fact, in our first high resolution ChIP study, we did CMYC, P53, and SP1, and Kevin Struhl did the ChIP. And I asked him why he picked those, and there was no biological reason. He's just, other than, he said, P53 won't bind very much, SP1 will bind medium, and CMYC will bind a lot. And that was purely based on not only their known binding information from previous studies, but the complexity of the motif. Just imagine if the motif is a few letters, you'll get lots of binding. If it's medium complexity in letters, you'll get medium binding. And if it's a lot of letters, like P53, it's 2 heterodimers, you get very little binding. So that's just something to keep in mind too before you chuck a data set. That's all I wanted to throw out.
Yeah, thanks, Evan.
I want to say something too, because I have some data sets in there. So the like, you have to be careful when you're looking at just the binary red and green, because like, for instance, for AR, if you don't have the androgen hormone in there, you may have like 4 of the things be red, which we have for some estrogen receptor ChIP-seq. But the data is great. It's just, the factor isn't bound because there's no hormone. And like, you can imagine something not being expressed and have a really great ChIP-seq. but it's just not binding anywhere because that's the biology. So I don't know. I just want to throw that out there.
Yeah, I think both are very great points. So I think I mentioned the last time that we usually use a cutoff of number of peaks identified from a ChIP-seq data as usually as a 1,000 or 2,000. So based on the consideration that in most cases, even the motif is very long and very complex. In the size of the human genome, human or mouse genome, the chance of having enough, if the transcription factor is actually function or active in that system, there are usually, that's purely dependent on empirical experiences, there are usually at least over 1,000 or 2,000 binding sites from the entire genome. So well, of course, assuming that the transcription factor is active, that's not the case that Mike mentioned, that if the biology, in the case that if the hormone receptor is not activated, in that case, there won't be many binding sites. We're basically almost like a background, but not necessarily, but it still have some binding in there. But if the transcription factor is very active, that imagine that we have over 20,000 genes in the genome, then in most cases, at least several thousands of binding sites are normal. But in case if the sequence motif is relatively short and they can have a lot of occurrences in the genome, it could be orders of magnitudes more binding sites than thousands or tens of thousands or even hundreds of thousands in this case. Yeah, so in general, it's just want to give an idea of what the ChIP-seq or the ChIP-seq data look like and what the possible transcription factors, the transcription factor binding profile will look like for different factors in different biological conditions in the genome. So yeah, so this is something I want to briefly mention. This is a tool that my lab developed based on the large amount of collected ChIP-seq data for nearly 1,000 different transcription factors in the human or mouse genome. So we built up this tool called BART, standing for Binding Analysis for Regulation of Transcription. So once we have all of the ChIP-seq data, binding information for different factors in different cell types, we can build up an algorithm to infer or to predict what the possible regulators, transcriptional regulators that are function to regulate a given gene set, a known gene set. So using a gene list that are usually from a differential expression from a transcriptomic analysis, that we can use this tool that purely based on the ChIP-seq data to predict, to tell you what are the most possible, most confident transcription factors that are function to regulate the expression of the genes in the input gene list as compared with other genes from the genome. Now this tool can also help you analyze if you have a ChIP-seq data set, if you can use this as an input for all the sequence reads, mapped reads as an input, it can generate the, compare the genomic profile of your input ChIP-seq data with the genomic profile of the TF ChIP-seq data from the public domain and tell you which transcription factor looks more similar, likely to be the factor have the same genomic binding profile as your ChIP-seq data. So if you get an input of some known transcription factor, they will, as a positive control, you're most likely to get that right transcription factor on top of the prediction list. And this tool also have a web interface called BART Web that you can give it a try to upload either a gene set or a ChIP-seq data. So, oops. Yeah, I think these are all the online resources and the tools that I want to cover for the lecture today. So in the end, I want to mention several limitations of a ChIP-seq experiment that you either do a study design or do the ChIP-seq experiment, or to do analyze the ChIP-seq data. That there are some points that we always want to keep in mind about the limitations. So ChIP-seq experiment is almost highly dependent on the availability and the quality of the antibody used in the experiment. So there are, although nowadays that for most transcription factors, there are ChIP-seq data available, which means there are antibodies available. For some other factors or other proteins that we're interested, the lack of high quality ChIP-seq data might be limited, might be due to the lack of high quality antibody in there. So if we don't have antibody, ChIP-seq can, even if you do ChIP-seq data, even if you do ChIP-seq experiment, there's not much we can interpret from this if the antibody doesn't work. So the second point is that ChIP-seq technique is a semi-quantitative approach to study genome-wide protein in DNA interaction. So because of the experimental procedure that we need several steps of pull down the protein DNA fragments and purify the DNA, and need a certain level of PCR application for enrichment and for all the sequencing step involved. So the profile of the ChIP-seq or the enrichment of ChIP-seq mapped reads across the genome, or even including the peaks we identified, only tells the relative enrichment of DNA, protein DNA binding compared to the genomic background and or the input chromatin control of your experiment. So by saying that, I mean that for ChIP-seq experiment usually cannot detect a global change of a binding level. So there are some recent studies showing that some transcription factor like MYC in different conditions may have a global increase of the binding level and their activity, and were usually not be able to catch by canonical ChIP-seq data. Because when we do the peak calling or all the analysis, we kind of use the entire genomic background. If the entire background level are changed, we cannot detect this global increase or decrease. In that case, sometimes some spike-in control would be helpful, but in that case, the analysis will also be tricky. So number three, the ChIP-seq experiments usually needs many cells and could be difficult for some cases that including clinical samples, or in some mouse experiments in which we only have a limited number of cells from the tissue or from the sample we can collect it. And because this is the experiment is done from a large population of cells, which assume that all the cells in that population have the similar pattern of TF binding or epigenomic level, but in reality, the cellular heterogeneity may play a role, may be a caveat when interpreting the ChIP-seq experiment. So in the end, I just wanted to, we've covered a lot of information in the ChIP-seq data analysis, and we just want to summarize by emphasizing this take-home message. So I know many of you may not eventually become a computational biologist or bioinformatician, but why am I still learning these? All the sophisticated statistical modeling and different bioinformatics tools to analyze ChIP-seq data. So even we are not doing the data analysis on a daily basis, these learning some idea or some strategies for the ChIP-seq data analysis, as well as an analysis of any genomic data, I think they will help you to understand how the data were analyzed and how the data were generated. They may help you to improve the experimental design. For example, you want to whether you have a good quality antibody, whether the cell sample in a good condition, and most important, whether you have the correct or right control of your experiments. And I think I mentioned this many times, that quality control is the one of the most important issue we always need to keep in mind about data analysis. So I've already go through many quality control measurements, and they can help you to better understand the data and also better understand the experiment. So sometimes if you found that, especially if you give the data to the bioinformatics core facility, or then or your collaborator, or bioinformatics collaborator, then they tell you, after a while, they tell you that no, your data, your ChIP-seq data didn't work. And then in that case, you will, if you know the details, several details of the data analysis, you may be able to know that, to ask them back that what part of the quality that they can tell that the data didn't work. If, say for example, if the FRIP score is extremely low, you have a very few peaks codified data, you may want to go back and check whether the ChIP experiment works well. Otherwise, if the PVC is very low, you might be able to check whether you still have enough chromatin material before you build the library for sequencing. But if the quality is the bad quality happens from FastQC, then you may simply just talk to the sequencing company or sequencing service that you ask them to redo the sequencing if the other pretty much the sequencing didn't work. So that all of these can help you better integrate your experiment and experimental data. And last, as I mentioned today, that we can take advantage of these existing tools, especially web-based analysis platforms, to do the bioinformatics analysis, even without a heavy level of programming and coding, and take advantage of the data resources available there. Okay, so then I will just end here, and thank you very much. Then we can spend a few minutes to questions.
Yes, I have a question regarding fragments length.
Yes.
So can long fragments produced by nanopore or PacBio can be more helpful in the analysis, or the short fragments are better from Illumina?
For my own, I don't have experience of analyzing PacBio sequencing data, as most of the ChIP-seq data that I have seen are generated using Illumina sequences. I think it depends on what's the biological question you want to ask. If it's a typical transcription factor or histone mark in human or mouse or regular genome, you can imagine the protein-DNA interaction regions, or just happens in no more than several hundred base pair of a DNA fragment of that region. So in that case, using Illumina-based sequencing is already very well to solve this question, because Illumina-based sequencing, although they have a very relatively short sequence for every sequence reads, but they can generate a huge number of sequence reads in one run, hundreds or millions of them. So I don't think that's something that any other sequencing platform can achieve at this moment. And which is and the high sequencing depths or generate a large amount of sequence reads is extremely useful for ChIP-seq, specifically because we want to have enough enrichment on a lot of different regions in the genome. So that's, I think, for most ChIP-seq experiment, Illumina sequencing should still be the better approach than other sequence platforms.
Okay, thank you.
Yeah. Other questions or any questions regarding the assignments or the lectures? Fine. So I want to point that I uploaded an additional instruction documents for the rest for the question 4 to 6 about the assignment. So I think several of them contains several different options, several different approaches that you can take to generate the data, to perform the analysis. But please do let me know if you encounter any difficulties or any questions when analyzing this data in the assignment.
Changzi,
Yes.
thank you for that. I want to throw one thing out. Yeah, I see John Stubbs. Post questions to Slack. Yes, do it all. Slack, contact Changzi if you have any questions. Yeah. Absolutely. Please don't be shy. You know, we all became instructors of this course together to help you learn to analyze the data. Okay.
Indeed, and I want to mention that this is the first time that I give the instructions about this materials, and I really want to hear your feedback about what the level of data analysis, how deep what do you feel about this work, and whether it's helpful for your play with the data, analyze the data, or even integrate the data.
And the key is, like, if you get stuck, just go on Slack, ask a question, don't be shy. Remember, the assumption was that you started from scratch, and there are many students like that never knew Unix, never knew R. You just had half a week of Unix, half a week, and 1 week of R. That's not much, and bioinformaticians know it. And then now you're jumping into ChIP-seq. So please, don't be shy about asking questions. No question is dumb. I'm just going to say that outright. There are more experienced students. Changzi can help. I can help. Others can chime in. And if you feel, and if it's the way you feel, you can personally contact Changzi and ask him. So you really should be able to get through the assignment. I also wanted to throw out, Changzi, this was pretty funny. When you were trying to recollect about ENCODE, it's 17 years old. Wow. So I was in ENCODE pilot.
The pilot also started, I thought that's
2003. Yeah, it's been going forever. So it turns out, this is just kind of a fun fact. I'm pretty sure it was our science paper that started ENCODE. So it was our unbiased characterization of transcription, and what's his name? Oh my God, I'm blanking for the head of the NIH. What's his name again?
Collins.
What is it?
Collins?
Yeah, Francis Collins. Thanks. I just blanked. So Collins was the head of the NHGRI at the time, and he contacted Tom Gingeris. And basically, you know, the way this mafia works is he asked Tom to help him write the RFA, and others that were at the bleeding edge of doing genomics. But I just wanted to throw that out. It's 17 years old.
Wow. So that's 2003, right?
Yes, 2003.
So that's right after the Human Genome Project's completed.
Yeah, so so remember, tiling arrays gave you nearly as good data
Yes.
not as good, but nearly as good data. But it was ridiculously expensive because you needed a lot of chips.
Yes, at the time. Oops. Okay. I think Stefan muted. Any other questions? Yeah. If not, yeah, just do shoot me an email or post on Slack for any questions you may have. And otherwise, have a great weekend and stay safe and healthy and enjoy the rest of the lectures. Thank you.
Thank you.
Thank you.
Thank you.
Thank
Thank
you.
you.
Thank you.
I'm sorry, I have one
Yes.
question more.
Sure.
If someone are not a student, like a audit, can do a homework also and send you?
I'm sorry?
If someone is not a student, like a audit, like it's not involved in the, it's not a grad student.
You can still, yeah, you're still welcome to do everything and let me know.
Okay. Yeah. Thank you.
I think the only difference is that you won't be given a grade to give a score.
Yeah, exactly.
Don't
Okay,
worry.
thank you.
Sure.
Thank you.
Okay, thank you. How to stop share. Stop.